Hello everybody, in this video lecture we will cover extensions from the law of inheritance. This is a PowerPoint that I'm gonna use. Let's go ahead and begin. So Mendel's experiment with pea plants suggested that first, the two types of units or alleles exist for every gene. That means for every gene that you have two alleles. Second, alleles maintain their integrity in each generation, no blending. And in the presence of the dominant allele, the recessive allele is hidden with no contribution to the phenotype. So on this picture below, you can see a pair of homologous chromosomes. And in the textbook, they color one chromosome blue, kind of like tell you this chromosome came from your father, and they color another chromosome in red. So this chromosome, this part of the pair came from your mother. But those are homologous because, look, they have the same shape, the same length, they have centromere in the same location. And what is more important, they have the same genes in the same locus. Locus means kind of like address, right? Where exactly it's located. So at the same locus, they have same genes. Now this gene is for a flower color. So if I tell you flower color, you don't know, am I talking about white color, red color, orange color, yellow color? So gene is kind of like this. Gene just tell you that's a gene for color. Alleles are alternative version of a gene. So this allele is for purple color and this for white color, right? So because you always get a pair of chromosomes, right? That means each gene will be, present twice, right? So you have this gene and this gene. Well, sometimes this is where confusion comes from. They use allele and gene kind of like the same. It's not really the same, but I mean, I hope you guys get understanding that gene can come in the multiple alleles, right? Same gene can be represented by different alleles. That's what it is. So because you always have, let's say, this gene for hair color and this gene for hair color, you can have the same alleles, brown color, brown color, or you can have different alleles. But the point is that it's always two alleles exist for every gene, right? And they maintain the integrity, and one can be dominant and other can be recessive, right? I hope it makes sense. Now, some patterns, however, of genetic inheritance are not explained by Mendels law. Much more complexity exists, and however, a fundamental principle of Mendelian genetics still hold true. Now look this example here. Red flower, white flower. Well, if red is dominant, what we're supposed to have in a second generation? Well, we're supposed to have all red flowers, right? But how it is possible that we have pink? Do we have some blending? No, we don't, but that's a variation of Mendel's law. Or you might have a flower that has both red and white color. No, how this can be explained by Mendel's, right? So this example shows us variation of Mendel's law. Now, what does non-Mendelian? What does non-Mendelian mean? It means that not all traits are as simple as dominant and recessive. Some traits, they have more than just two alleles or variations. Some traits have two dominant alleles. Some are passed only via sex chromosomes, and some traits are a result of hundreds of genes being expressed. Like this calico cat. Just Mendelian genetics cannot explain how you can have all these three different colors in one organism, right? If black is dominant, okay, it should be black. If white is dominant, should be white, right? So how we can explain these patterns? That's we need to use non-Mendelian genetics. But non-Mendelian genetics is still supports Mendelian genetics. It's just more complicated. Mendelian genetics, over many experiments, we accepted that that's reliable hypothesis and laws that Mendel provide us with. But of course, life is more complex than just simple dominant and recessive. So one example of non-Mendelian genetic is incomplete dominance. So this is a flower snapdragon, and if you have P generation, that is purebred, red and white flowers, so big R, little r, if you apply Mendels Law in F1 generation, because red is dominant, you should have all red flowers, right? However, for this particular organism, in F1 generation, you will get pink flower. It's still gonna be heterozygous, capital R and lowercase r, but red color is not completely dominant. That's why it gives this pink appearance. But how we know it's not blending? It's not like you have red paint and you mix it with white paint and then you have pink paint and you don't have red and white anymore. There is no blending really in genetics because those genes for white color, they have their integrity. They still there. They just not completely expressed. They're not lost. There is no blending, and you know it because in F2 generation, this white reappear again. Just to be white for this flower, you need to be little r, homozygous recessive. To be red, you need to be homozygous dominant. And if we have hybrid, heterozygous, it will be pink. So that's what incomplete dominance is. F1 hybrid have an appearance between the phenotypes of the two parents, right? So it's in between. Here's the example of incomplete dominance in people. Hypercholesterolemia. Hyper means high. Cholesterol means a high cholesterol level, and lemia means in blood. So those people have high cholesterol in their blood. Now, look, when you have cholesterol, and this is show LDL, this is what we call bad cholesterol or low density lipoproteins. So that's a bad cholesterol in your blood. So if you want to remove this cholesterol from blood, actually the liver cells, so this represents liver cells take it from the blood inside the cell. So that's why you don't have high cholesterol in blood. But to take this LDL inside liver cells must have receptors, special receptors. So now look, in a homozygous dominant, so that's a person without this genetic disease. Person has plenty of these receptors. So it takes this bad cholesterol inside the cell out of bloodstream. Now, if somebody has one gene with a mutation, heterozygous, this person has less receptors, so it will cause mild disease. And if it's little h, homozygous recessive, that means both genes have a mutation, this person doesn't have any receptors. So cholesterol level is really high, so it's a severe disease. So this is incomplete dominance. Because look, if big H would be dominant, completely dominant, this person, it's dominant, would have the same number of receptors. But no, this person has kind of in between, right? You see, normal and severe, and this person in between, right? The same as here, red and white, and this flower is in between. The same here, normal number of receptors, no receptors, and heterozygous or hybrid in between. So this is incomplete dominance. So heterozygous have blood cholesterol level about twice normal, Heterozygous, twice normal. Homozygous have about 5 times the normal amount of blood cholesterol and may have heart attacks as early as age 2. So that's this person over here. Codominance is another alternation of Mendelian inheritance law. When both alleles for the same characteristic are simultaneously expressed in the heterozygote, that means they both are dominant. Example of codominance would be blood group A, B, and O in humans. And it's also multiple alleles because look, we don't have only A and B, we have A, B, and O, 3 alleles, and 2 of them are dominant. A and B, they both are dominant, right? So this is how it's abbreviated. They put this little I, this is recessive allele, capital I are both dominant alleles. So A and B are both dominant. So I for either glutinogen, right? It's another term for antigen. But we can just pretend that, oh, well, too easy, if we want to look easy on this codominance with blood type, we can just assign O is recessive allele and A and B, they both are dominant. So now look what we have over here. If person has big A genotype, then blood type gonna be A. If person have big B genotype, then blood will be big B. But if person has A and B, neither one is dominant, so the blood type gonna be AB. And the person that has little I, that blood type gonna be O. Right? In physiology, you will go into details of all these antibodies and antigens, but for purpose of this class, we just wanna remember that codominance, when both alleles are dominant, right, and they both can be expressed. An example would be blood type inheritance in people. So here, cross a person with blood O type with a person that has blood type AB and give phenotypic and genotypic ratio. So again, pause this video and try to solve this problem. Okay, so this is what we have. This is person little I, right? This is a person with blood type O, and this person with blood type AB. And it's simple, really like monohybrid cross. So you fill in Punnett square, and this person has blood type A, B, and B. So genotypic ratio 1A, 1B, phenotypic ratio 1 to 1, type A, type B. Look, the person with blood type O cannot even have child with blood type O if a partner is type AB, because both A and B are dominant. A type B blood male and type O blood female never produce a type O blood child. Is this possible? Why or why not? So we have type A, type B, right? Parents, and it says they can never have a child with blood type O. Is it true? Well, pause the video and try to figure it out. Well, so we have, let's see, type B blood male. So if this is type B, this person can be big B, or big B and little I, right? So this is 2 possible genotypes over here. So if we will take the person with blood type O, little I, and type big B, then yes, child cannot be type O. But if parent is heterozygous for blood type B, then they can have kids type O. Let me get a pen. Look, I'm gonna use it a little bit different. I'm gonna say that one parent is OO, right? Just easier. And another parent is type B, right? Well, but what type B? It can be like this, right? That's a type B. Agree? Or one parent type O, so it's O, O, and another parent is type B, but that's gonna be B and O. So instead of I's, right, instead of this I, you can just use O because O is recessive. Now, if I have these parents, over here, how my Punnett square gonna look like? Right, here we go, right. So what I need to do, I need to split these O's, right? So here's O and O, and I need to split this B and O. So here's my B, and this is my O. And here you fill in B, O, B, O, right? And what do I have here? O, O, and O, O. So now look, if one parent is, oh my goodness, I'm losing this. This is so tiny, I cannot see it. Okay, here we go. If one parent is this type of B, then no, this parent with another partner with blood type O can never have kid, blood type O. But if I have this parent, so actually chances are 50-50. 50% the child will be type B and 50% the child will be type O. Now, polygenic inheritance is another exception of a Mendelian inheritance and is editing effect of two or more genes on a single phenotype. So look, these people have all different height, right? So how possible that we don't have like short people and tall people? If tall is dominant, you know, we should have like tall people and short people. Why we have people of all different height? Because your height is determined by more than one gene, several genes. So over here, short allele shown like this white color, and tall allele is a black color. So we have 3 genes. 3 genes will give you 6 alleles. So now if your parents give you all 6 short alleles, then you're gonna be very short. If your parents give you all 6 over here tall alleles, you're gonna be very tall. And kind of like everything in the middle has all people have all different height. So this is called bell-shaped graph. So any trait that shows this bell-shaped graph will be very few people of one extreme phenotype and another extreme phenotype, and most people will be in the middle. Then it's polygenic inheritance. Example of polygenic inheritance in human includes height, skin color, and hair color, and eye color, right? So everything that we have, look at this eye color. We don't have just brown and blue. We have light brown, dark brown, hazel, we have dark blue, light blue, right? All these different colors because we have more than one gene that determine eye color. Our skin are also all different. Why? Because more than one gene that determines skin color. Link genes. Link genes are located close together on the chromosomes and inherited as one unit. So these link genes are not inherited as independent assortment. For example, red hair and freckles, because they located so close on the chromosome, most people will inherit both red hair and freckles, right? Or have none of this. So linked genes are inherited together. That's why we call them linked. The only way to split 2 genes on the same chromosome is crossing over. We described crossing over in a meiosis lecture. So if you watch this, a meiosis, that is sexual reproduction division, it will explain you what crossing over is. So look over here, that's 2 homologous chromosomes, right? And again, blue from dad, red from mom. And on the blue, we only have dominant genes, and they are linked because they located on the same chromosome. Here we have all the recessive genes, and they also linked. So really, I kind of need to, well, the offspring should receive either all dominant or all recessive, because you inherit the whole chromosome. But this is crossing over when homologous chromosome can split their fragments, and then we have recombinant chromosome, so we can actually separate those linked genes. So the baby that inherits this chromosome will get 2 dominant and 1 recessive gene, right, or allele. Excuse me. So no crossing over, keep parental combus, and crossing over, get new combus, recombinant gametes. So those are linked genes. They can be on any autosome, any chromosome. But when linked genes are on X or Y chromosome, we call it sex-linked genes, and they produce sex-linked traits. In humans, as well as many other animals and some plants, the sex of the individual is determined by sex chromosome, one pair of non-homologous chromosomes. When a gene being examined is present only on X chromosome, but not on Y chromosome, we call it X-linked. So you can see X and Y chromosomes are not homologous. Y chromosome is way smaller and it's missing a big part of the X chromosome. So this gene A, B, and C only found on X chromosome. That's why it's X-linked. So, so here's our females. When we're looking at sex-linked traits, we always use X's and Y's. So XX for female and XY for male. Now, female can be homozygous dominant, homozygous recessive, and heterozygous, right? That's a carrier that carries this some defective allele. Well, allele with mutation, right? This example, hemophilia. But first, when we're talking about sex-linked genes and when we're talking about genetic condition, we use the first letter of this condition, and if it's recessive, we give it lowercase letter, right? And for normal, so here's, remember how we usually look at the dominant trait and use the first letter of dominant trait, so we should use N. But for genetic disorders, we use letter of the disorder. And we use little h or lowercase h because hemophilia is recessive. So female can be normal or hemophiliac, or she can be a carrier. Now in males, we don't have a carrier. We can only have normal person or hemophiliac. And then, well, this is homozygous, this is heterozygous. What is this? This is hemizygous. Hemizygous means it only has one allele for a gene. So hemizygous. Crosses involving sex-linked traits often give rise to different phenotypes for different sexes of offsprings. So if you're looking at sex-linked traits, then the ratio for females will be different from ratio for males, right? So over here, what we're looking here, let me see, red eyes, right? So, so white eyes is a mutation, and white is recessive. So we put little W's here. So this is female and this is male. Male has a red eye. And again, we're separating this X. So here's our female, X with little W, X with little w, and this is male gametes. X with capital W and Y. And then we just fill in Punnett square. And you can see that female, we have what? 100% red eyes. For male, we have 100% white eyes, right? So you see ratio is different for males and females. So in humans, the alleles for certain condition, color blindness, hemophilia, muscular dystrophy are X-linked. Females who are heterozygous for these diseases are said to be carriers. and may not exhibit any phenotypic effects. X-linked traits appear more frequently in males than females. So here's a genetic problem. What type of offspring would a colorblind man and woman who is carrier for colorblindness have? So you can pause the video and try to do it yourself, and I have answer for you. Okay, so again, we're talking about color blindness. Color blindness, so we use letter C to represent, and because it's recessive, we use little c for color blindness and big C for normal. So female is carrier, so it's big C, little c. She is not colorblind. And male is colorblind. So it should be kind of like little c, but because it's X-linked, the female gonna be, look, XX, they both carry those alleles. So X, capital C, X, small c, but for a guy, it's homozygous. It's X with little c and nothing on Y. And that's why it gives you completely different ratio over here. So you put a female here, her alleles, and guy alleles over here, and you fill in the Punnett square. And look what you, and for X-linked, you need to give kind of 2 ratio for female and male. So this is female, this is female. So one female over here is carrier and another colorblind. And for guys, one is normal and one is colorblind, right? So no carrier in male. Epistasis. Genes may also oppose each other, with one gene suppressing the expression of another. In epistasis, the interaction between genes is antagonistic, such that one gene masks or interferes with the expression of another. The alleles that are being masked or silenced are said to be hypostatic to the epistatic alleles. So we have epistatic, epi means kind of on top, hypo means below. So one is epistatic, another is hypostatic. So the one that is silenced is hypostatic to epistatic alleles that are going, that are doing the masking. So example of epistasis would be albinism. So albinism is an example of how one epistatic gene blocks the production of pigment in skin, hair, and eyes of animals and humans. So let's look over here. That explains us this epistasis in dogs. So this is Labrador for color, and black is dominant and the brown is recessive. So far it's easy. B for black, little b for brown. So here's black, here's brown. Well, how about this yellow? How about this yellow Labrador? How we come up with this? Now, because we have another allele, and this allele for melanin deposition. So this dog can synthesize melanin. This dog can synthesize melanin, right? In a less amount. This dog maybe has melanin, but it's not deposited in hair, eyes, and skin. Now, if this dog has little e, so no melanin deposition, so it doesn't matter what the first 2 alleles are. It can be big B. So maybe this dog has homozygous for black color, big B, but because of this epistatic alleles, these epistatic alleles inhibit the melanin allele, and outcome will be yellow Labrador. So the recessive genotype of little e will cause no melanin deposition. Thus, the resulting fur coat will be yellow even when B or little b alleles are present. So this dog can have little b and still be yellow. This dog can have big B and still be yellow. Or this dog can have big B, little b and still be yellow. Now these 2 dogs, if they have at least one normal allele for melanin deposition, right, then this first allele will be expressed, right? Either it's going to be dominant or it will be recessive. Hope it makes sense. So let's see how much is left. Okay, this was our last slide. And in this video lecture, we covered extensions of the law of inheritance, and we talk about, I'm just going to remind you very quickly, incomplete dominance. We talk about codominance, that when we talk about blood type. Also polygenic inheritance and link genes, right? We're talking about link genes. We talk about X-linked genes over here, or X-linked traits and X-linked genes, and we talk about epistasis. Thank you for watching, and I hope it was helpful.